I will talk about synchronous and asynchronous computation in the brain from the neuroscience perspective, but also from the theoretical neuroscience perspective. So I will also show you some kind of modeling results. And let me just briefly summarize. I mean, usually in computer science, synchronous in a synchronous logic, you basically have a periodic clock signal, and the kind of the computation is always triggered by the clock signal. And of course, asynchronous computation is kind of more event-triggered, is basically characterized by the absence of a clock signal. That's just very roughly these two different categories. And now let's look in the brain network, and we have already seen here this network before. So this is basically, let's see. So what you see here is the result of an analysis of actually cortical connections, and it's actually using the COCOMAC database that was actually started by Rolf Kötter, a former colleague of mine. And this is actually work by Dharmendra. And as you can see here, and you cannot really see it, here's all the cortical brain regions, and you can see kind of the bundles of fibers going between these brain regions. And this is kind of the coarse view of the network that we are dealing here with, of the computation network. And on the fine scale, of course, and this is also something you have already seen here, we have the six-layered microcircuit of cortical of cortex, and you basically have very rich local connections here within these cells. And Valentin Breitenberg actually and Almut Schütz, they kind of started kind of a field called quantitative neuroanatomy. So they really started to really count in detail, you know, how many let's say in the cortex of the mouse, what is the percentage of local excitatory connection versus these far-ranging fibers. And the result of this analysis is basically that most of the connections are local. And most of the connections are excitatory. And so roughly speaking, we have here a network which is very different from a deep learning network. So we have a very strong local connectivity, and it's an excitatory connectivity. And so the question is now, if we look actually in the physiology, do we see, are there hallmarks of clock signals? And I mean, a clock signal is a periodic signal, and it's very interesting that actually neural oscillations or neural rhythms were observed, not in the last century, but in the last but 1 century. In neuroscience, we have this funny situation that we stand on the shoulders of giants which come from the last but 1 century. And basically, it started in 1875, and then Adolf Beck really examined, without having access to electronic amplifiers. He found basically ongoing rhythms in brains and also event-related potentials, basically. And of course, since then, rhythms have been observed all over. They are in the periphery, they are in the retina, they are in cortex. There are actually on many different temporal scales. There are slow and fast rhythms, and in the 2000s it became very popular to think in these rhythms in terms of communications between brain regions. And so there's actually this interesting concept of the oscillatory hierarchy that kind of talks about nesting of different rhythms, and mainly for the purpose of communication. And so yeah, there are rhythms, but is there one clock rhythm that actually governs how it is computed in this network? No. It's a funny situation. So this is actually data from the lab of Emery Brown, and he's also an anesthesiologist. And what he actually did here is that he put basically a person to sleep with propofol and woke the person up again. And this vertical bar is basically here basically where the consciousness went away and came back again. And you can see that actually under anesthesia, we see the strong and very global rhythm in the EEG, but not as soon as the brain is in a conscious state. So these strong rhythms are not hallmarks of these strong global rhythms that could look like clock signals are definitely not hallmarks of the regular operation of the brain. So let me therefore focus now on a brain region you have already heard about quite a bit, and this is the hippocampus. And this is actually in Darmendra's diagram. This is just here, this little sliver here. And here you see a blow up. And so the hippocampus is very important for kind of laying down episodic memory. So it's a memory region, in particular for first contact with the memory content. And it's also very important for navigation. And if you recall from a single cell here in CA1 or CA3, what you actually find is neurons that have place cells, which means that if you actually let a rat run through this hallway here and you make a dot of a certain color for one neuron, you will actually see that, for example, here this pink neuron fires only in this part of the environment of the animal. And so you have other neurons that then eventually tile the entire environment of the animal. And so that is what you see on the single neuron level. But of course I have chosen this region because there is a very strong oscillation in the hippocampus during active behavior of the animal, so during active voluntary behavior. So if the animal walks through the environment, if the animal presses a lever or does stuff, then you actually see this rather regular rhythm here. It's about 10 per second, 10 hertz. And this is called the Theta rhythm. And it's one of the strongest rhythms actually in the brain. You can actually see it if you just take an electrode and go close to that region, you actually will see it. And so now the question is, of course, how does this structure the computation in this brain region? And now here's again actually a slide you already have seen. And what you see here on this side is that there are actually, so this is now basically multi-electrode recording from the hippocampus. So there are these colored electrodes here. And in the data, there were certain spike trains. So on each electrode, there were actually certain spikes detected from neurons, and they are basically represented here just as rastergrams. And here they are solved by electrode. And you can see in the animal, and you can see here, this is its theta rhythm. So the animal is walking through the environment here. And you can see here that cells are firing. There are the green cells, the red cells, and so on. You don't see very much structure. If you look here on the right side, then all of a sudden you see here the structure, which is also emphasized by these ellipses circled around them. So you see these so-called cell assemblies. So you saw this in Christos's talk yesterday. And so it seems, and so you can also see that actually these bursts of activity in different neurons, and they are here spread across the different electrodes. So they are not localized, they are spread out. You can see that there are more precisely timed, basically, than what you would expect so the animal compared to this very short stretch of time, which is maybe a second or so, the animal has not moved very far in the environment. So just by the outside pacing of the environment, you wouldn't expect a timing which is so precise here. And so therefore, seems here from this picture that indeed the theta rhythm organizes the representations into these cell assemblies in the hippocampus, and that you basically can only see this after you reorganize the pattern, basically the spike trains from here to here. And what actually Ken Harris, who produced this data, or actually who analyzed the data in 2003, he actually took it out of the trash bin of the Busaki lab. What he found is actually, so what he used here to resolving the data is kind of an algorithm that compares the similarity in time of the different spike trains. So now, what kind of computation could you locally do with this? And so this is actually here the result of a simulation model that we did a long time ago, that Thomas Wennickers, around 2000. And so these are basically a bunch of conductance-based neurons, two-compartment neurons. And basically, we kind of stored local cell assemblies in these in the network. So basically, in the synaptic structure, there are now certain patterns that are like attractors. And what you can see here, that in each of the oscillation cycles, here you can see it very clearly. So there are here these a little bit earlier coming neurons, and they are stimulated by the outside. And then you can see the other 5 here coming on, and this is now the network effect. So you can see that these oscillations could actually organize content addressed memory recall cycle per cycle. And so this is basically, so this is now all saying that, you know, locally, these rhythms could actually organize and even actually synchronize certain computations and kind of form representations that are more synchronized in time than what you would expect, for example, by the changes in the outside in the sensory signals. And it has been shown by physiologists like Randy Bruno and so on that actually this focusing in time could be very important to be actually for actually synaptic inputs to be heard in a subsequent area. Because, and here I come back to what I said earlier, because most of the connections that a neuron actually has in a cortical area is recurrent connectivity. So it's not so clear, you know, how an LGN input 2V1 can be heard is not as clear as you would believe if you see the textbook picture. Because somehow V1 has to figure out that among all these recurrent inputs, there is actually some input from the input now, which is important if a sensory input is important. So now let me kind of go on to another situation, and this is now basically going beyond synchronous computing. So this now shows actually just a cartoon of an idea that actually Pascal Fries put forward, which is more about, it's an idea how actually routing could be performed in the brain, and routing in the sense that basically functional networks are built. So a brain area by itself can do almost nothing. It's always basically the functional network for a certain task that needs to collaborate in order to perform a function. So how to basically set up a functional network almost on the fly is very important as we switch from one task to the next. And so his idea was, so we have here 3 brain areas. These are now entire brain areas, not neurons, A, B, and C. And A and B have an oscillation which is coherent in phase, so the phases are almost the same, and C is out of phase. And his idea was now that often these rhythms, they also go along, they basically also modulate the excitability of neurons locally. And so his point is that these 2 guys are in a good position to talk to each other because actually their signals arrive on the other side when actually there's a maximum of excitability and therefore elicit spikes here. Whereas B and C have a hard time to communicate. And this is called the communication through coherence hypothesis that actually Pascal has produced some data to actually support. Now, Okay, so now this is now, of course, a way how actually you already need kind of an asynchronous circuit to actually set up these functional networks by actually decoupling some networks and putting them, decoupling some regions and putting them out of phase of the other. But the picture becomes even more complicated if you look at conduction delays. So this is an old graph. This is basically from Harvey Swadlow's work, and this shows you here. So on the x-axis is actually in milliseconds delays, conduction delays, measured by antidromic stimulation. Over basically it's just a histogram. So how often he just in many cell pairs he measured the conduction delay. And this is actually, I think, through the callosum. So this is not a local connection. This is really a kind of a long-range connection in cortex. And what he finds here, well, there's a peak maybe here around 7 milliseconds, but you have a pretty heavy tail here of very, very slow fibers. And it is not clear that if two neurons fire together, that actually their signals arrive at some neurons they synapse to at the same time, given that you have this dispersion here of conductions. And therefore, Eugen Itzikevich and also Busaki put forward that maybe what really counts is not synchronous firing, but synchronous arriving of basically synaptic inputs. And Itzikevich calls this polysynchrony, and actually Busaki calls these synapsemblies. So it could be that at least for if long-range connections are involved, that it's not just about synchronous firing, but it's more about actually synchronous arrival of spikes. And this could actually be, for the physiologist, a quite different picture. So we actually studied a little bit how if you actually put in these, this is just the same picture here, Harvey Swadlow's distribution of conduction delays in a simulated neural network, again, conductance-based neurons. And let's say you have here two brain areas that have an excitatory, inhibitory, and two inhibitory pools here. And so here in the middle are the long-range connections. So these are the connections that actually have these delays. And if you just simulate such a network, then actually you find that oscillations, at least in the gamma range, which is the higher frequency range, 52 to 70 hertz or so, that that what you see is you don't see in-phase synchrony between these two regions because the delays are just on average too long. But what we could show is actually that if you now assume that there's STDP learning rules, that then very quickly you can actually establish a zero-phase synchrony between these two regions. And so, but this is definitely conduction delays can, at least for long-range connections, definitely cannot be neglected if you think about how actually the communication takes place in the brain network. So now let me come back to the hippocampus. So in the hippocampus, there are these place fields. So there are two neurons, blue and red, and in here are their spike trains. And you can see that the animal walks here from red to blue, and red fires here, blue starts to fire here. But if you now actually draw here, and this is just a cartoon. If you draw here the theta rhythm, and you note here the relative phase of let's say the red spikes and the theta rhythm, you actually find that they actually advance in phase as you go from here to here. It's just maybe a little bit small effect here, but basically this relative phase turns out to tell you something of where in the Blaze field the animal is, and that's called phase precession, was actually discovered by O'Keefe and Retche. And so here we have now a situation where actually this relative phase becomes important. And there's also models that actually showed, so that was a very simple model actually by John Hopfield in 1995, and he showed basically that if you have actually, let's say, a sensory input, which is just rate coded here, high rates here, less so here, higher rates here again. And now you have actually an oscillating neural network where, again, the oscillation determines how excitable a neuron is. Then, of course, you will see that the strongest excited neurons fire first. And that basically in the latency of these spikes, you have basically an encoding of the strength of the sensory input. And so this could be a way how actually now this local clock signal does actually more than just organizing the timing and maybe the representation of cell assemblies, but could really have a function here in, for example, transforming a rate code into a latency code and things like that. But of course, for a long time, people believed that actually this rhythm that you see here is the same, so that it's the same everywhere in the hippocampus. And that, of course, would then make it easy to think about such schemes of what actually this oscillation could do for you computationally. However, in actually 2009, Thanos Yappa's lab actually showed that in the hippocampus, the theta rhythm, which is really one of the strongest rhythm in the brain, is actually not has not the same phase independent of space. So this shows one depiction of the hippocampus where this is the spatial, the septotemporal axis. Don't worry about this right now, but basically this is actually a figure from a follow-up paper from the Busaki lab, but they placed electrodes there and basically measured here the so on the x-axis, you have the relative distance here from this 0 point along the septotemporal axis. So this is just this axis here, is on the x-axis here. And on the y-axis is basically the phase angle. And so you basically can see that as you wander along this axis, the phase angle just goes up by basically 180 degrees. And so this means that actually this phase angle is a function of space. And if you look at it in time, you actually see this. And this is basically where a project with actually Gautam Agarwal started, a former postdoc in my lab. So we basically asked the question, okay, so you see basically here, this is the anatomy of the of CA1, and we had an 8x8 electrode array, and this is now again Busaki lab data. An 8x8 electrode array was here implanted, and so basically every point here on this plane, it's an 8x8 grid. And this is basically just depicts the signal strength as a function of time. And this is, again goes 10 per second. And so basically what we asked with Gautam was basically, well, we know that there is relative place information encoded if you know the spikes and you know the relative phase to the theta rhythm. But can we actually, if we have a spatio-temporal structure of this hippocampal wave, can we decode the behavior of the animal? Can we decode where the animal is in the environment? And this is kind of, in the hippocampus, is the decoding question difficult because there is no topography. So nearby place cells are not nearby in the hippocampus. So therefore, these are very mixed signals, unlike to V1 and A1, where often even if you measure on one electrode and you do not sort spikes, you get a very nice preference in orientation angle or in the feature. We were, though, relatively optimistic based on some actually theory work that we did on actually compressed sensing, which can basically be applied to the situation that you record with a relatively small number of electrodes in the tissue with a relatively large number of neurons. And then, of course, the second obstacle to decoding the wave is basically that there is an intrinsic dynamics. There is this rolling wave through the tissue, which by itself is not correlated with the behavior and therefore certainly not helpful for decoding. And for that, actually, it helped that the senior authors of this study, Guri and myself, we kind of grew up building transmitters. And so we were very familiar with concepts of actually modulation and demodulation. And so basically what we did is we took, so this is just the raw signal. So here are the channels, and this is time. And so basically what we did is we took the first principal component of this spatial temporal signal structure and declared this as the carrier. And then basically did a phase demodulation using that carrier. And the result of the demodulation is here. And you see this is already a much slower signal, much more in line with the behavioral time scale actually of the animal. And what you see already here is these are now trial repeats over many runs of the animal to the right and to the left. And you see there is some resulting structure in the trial average, which already is very encouraging for decoding. And so I have to speed up, but anyway, so we basically could decode. This is basically using the spikes recorded from the same electrodes for decoding where the animal is, and here it runs left and right. And this is just the LFP structure. And so you can see here in this green trace that the LFP structure is as good as what you get with the spikes, and more interesting, if you combine the two things, you actually get an improvement. And we also applied unsupervised learning directly to these LFP signals, and we kind of found actually the independent components of this are actually, so these are now trial repeats on the y-axis, and again, and location on the x-axis here. And you basically see each of these colors correspond to a component of the LFP signal, which was highly selective to one location in the animal. And we have a complete set tiling the entire environment. It is just using unsupervised learning. And in the electrode space, all these components are extremely distributed across all the electrodes. So that's very different from a neural signal, from what you would see if you would do that independent component analysis actually on the high frequency spectrum that you use for spike sorting. So, and just this shows you basically that this works also in more complicated environments. Here, these different colors correspond to different blaze components extracted by this unsupervised learning. So to sum up, basically what I've shown you that basically the physiology and also the abundance of excitatory recurrent circuitry suggests that neural rhythms are a part of the brain dynamics and function. Oscillations, ongoing or induced, can shape cell assemblies, support learning, and set up large-scale functional networks. That's the more traditional stuff. The new data here basically kind of suggested that the oscillations often carry specific task information. So it's not just a clock signal, it's also not just a signal about the global state of the brain, but it is a signal that you actually can, for example, use to decode where the animal is at every point in time. So therefore, the suggestion is that the local field recordings simultaneous to spiking activity are important and can really help to understand how subthreshold population dynamics and spikes interact to produce computation. And so I think right now the field of neuroscience is very spike focused. So everyone wants to sort spike and look at spikes. One should not throw away all these signals that are in the low frequencies and that you can extract from the same electrodes that you stick into to decode spikes. And currently, neither brain theories nor neuromorphic models provide an insight about this interplay between actually spikes and these continuous population subthreshold events that we see in the LFPs. And I think that is certainly not would be a very interesting direction to actually get new ideas for neuromorphic computing. Thanks a lot for your attention.
Time for a quick question while Rajit sets up.
What you're implying when you say that brain theories have nothing about what LFPs are or they mean. I mean, it's been pretty mainstream that this is the sum or the weighted sum of distributed local driver currents with filtering and blah 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 in details. But why isn't that counting as brain theory respecting what LFPs are and what they mean?
Oh no, absolutely. There are, of course, a lot of theories what these LFPs, how there is work, some work on how these LFPs are composed and so on. But still, I think there is not much work that really relates. So, for example, how does spike-based computing look in the presence of these structures? I think there is not much work. I mean, I'm happy to learn if you have some work, but I think right now we don't have a good idea how these I would say these analog processes, these analog structures of the LFPs, they fit like a jig puzzle to the structure in the spikes. And we should understand this interaction between both, and I still think there is not much theory that tells us how we could use that for computation, in particular including the rhythms.
So do you think of the fields per se as being contributive to, or just as evidence of synchronous activity among regional neurons, or is that a longer question that we should put off?
Let's take it offline.